Probably 1 or 2 minutes are left. Today we are meeting after a long gap, and the topic selected for the today live speech is catabolism of glucose. Of course, the topic is significant for your coming examinations, and the catabolism, I selected the topic because you all are well aware for the topic in your previous classes. We well known that the catabolism is a part of metabolism, and if we simply say that the metabolism, what is metabolism, then anyone can say that the overall biochemical reactions occur within cell are collectively referred as the metabolism. Basically, the metabolic reactions are responsible for 4 functions. First one is to extract chemical energy from environment, either from sunlight or from organic nutrients. If the chemical reactions extract the energy from the sunlight, then we refer it as the photosynthesis, or the other form or other types of reactions which are responsible for the derivation of energy from the organic nutrients are referred as chemosynthesis. Of course, today we are confined to the metabolic activities of the glucose, and particularly it's the catabolic part. But I am going to supplement some necessary informations related with proteins and fatty acids also. Of course, we will discuss the rest parts related with the proteins and fatty acids in coming classes. Then it is the first work of the metabolic reactions. Second work is to convert exogenous nutrients into the building blocks or precursors of the biochemical macromolecules. It is the second function of the metabolic reactions. Third one is to assemble the biochemical macromolecules into precursors or into the macromolecules like proteins, nucleic acids, and lipids. Then The fourth functions of the metabolic reactions are to form and degrade those biochemical molecules required in a specialized function of cells. That means the metabolic reactions are responsible for these four functions of the living system. Of course, we discussed a lot of the bioenergetics and enthalpy and entropy parts in previous classes. So, some reference of that particular classes are also necessary here. And if we consider the metabolic reactions with respect to their function, then they are broadly divided into two parts. First one is the catabolism and the second one is the anabolism. The catabolic reactions are responsible for enzymatic breakdown of macromolecules into micromolecules. If the macromolecules are splitted into the micromolecules enzymatically, then we refer the reactions as the catabolic part of cell. And generally, the catabolic reactions are associated with release of free energy in the system, and the part of the free energy released during the catabolic reactions are conserved into the living system in the form of ATP, which we have discussed in the coupling reactions in my previous classes. The second part is the anabolism, and anabolism are responsible for the synthesis of macromolecules from micromolecules and increase its complexities. And again, these reactions are also associated with the enzymatic activities, because in the anabolic reactions, the organizational complexity is increased, then it is responsible for the decrease of entropy. Hence, it requires free energy. Because the entropy is decreased, it requires the free energy, and all these reactions obtain the free energy in the form of basically ATP. In some reactions, the thiol group is associated, which we are going to discuss in this class. Then they require the ATP for the anabolic reactions. These facts are a brief introduction before coming on the topics which we are going to discuss today. That is the catabolism of glucose or glucose catabolism. Here, I am saying that the catabolic reactions are responsible for the energy production, and carbohydrate is the major or main nutrient which we use for the production of energy. But we also obtain the energy from the lipids or from the amino acids, that is the proteins, in some specific conditions. So, we are going to discuss fundamentally on the catabolic activities of the glucose, but I provide some reference if we or if I think that is necessary for your coming examinations. Then, in the catabolic reactions, just I am saying that the macromolecules are required for the production of energy, and these three macromolecules are present, that is the in the form of the carbohydrates, in the form of the proteins, or in the form of the lipids. Then at first, before coming on the catabolic reactions of the glucose, the catabolic reactions are split in the three stages. First, the enzymatic breakdown of the macromolecules. At first, in living system, the enzymatic breakdown of macromolecules are occurred. That means the carbohydrates enzymatically break into the monosaccharides, particularly in the form of hexose, by the enzyme amylase. Second, the lipid breaks into the fatty acids and glycerol by the enzyme lipase. Likely, the protein also breaks into the amino acids in the presence of different types of the proteases. I am saying proteases, but the nature of the enzymes is different with respect to the different cutting sites in the protein, which we will discuss in the chapter of the enzyme. And when the building blocks are released enzymatically in the living system, they are firstly associated with the anaerobic respiration or anaerobic breakdown. And anaerobic breakdown is occurred in the cell cytoplasm. In the cell cytoplasm, and the end product is lactic acid. That particular stage is referred as glycolysis. Now, the end product of the anaerobic reactions in the cytoplasm, or the enzymatically digested lipids and proteins, are entered in the aerobic catabolic reactions, and the main aerobic catabolic reactions are we studied under the heading of the Krebs cycle. That means the catabolic reactions of the biochemical molecules are easily, we can separate easily the catabolic reactions in the three, under the three heads. One, that is the catabolic breakdown of the, enzymatic breakdown of the macromolecules. Second, the anaerobic breakdown of the monosaccharide. And third, the aerobic breakdown of the enzymatic end product of the lipid digestion or of the proteins, which for the lipid digestion and for the proteins we will discuss in the next classes or in the coming classes. But today we confined our discussion or our speech only on the catabolism of glucose. Then the first part, after the digestion, after the enzymatic digestion of the macromolecules, the first part is the anaerobic breakdown. or and anaerobic breakdown is occurred in the cytoplasm. The anaerobic breakdown of glucose is either glycolysis, or in the some cases the fermentation is also the uh, part of anaerobic breakdown. In some cases fermentation is not observed commonly, but the fermentation of a step is also the anaerobic breakdown of the glucose. If we study the glycolysis, and we all know very well that the glycolysis is also known as the Eden-Meyer pathway, because after it, the name of the discoverers, then the Eden-Meyer pathway. And if we study the glycolysis, then glycolysis requires 11 enzymatic steps. Total 11 enzymatic steps are present in glycolysis, and the end product of glycolysis is 1 molecules of glucose is responsible for the production of 2 molecules of lactic acid. That is the end product of glycolysis in the absence of oxygen or in the complete anaerobic breakdown. When the fermentation is observed, then the glycolytic steps exceeded 2 steps to form 2 units of ethanol and 2 units of carbon dioxide released from the glucose. Now we comes on the different steps of glycolysis. For the better understanding, the glycolysis is divided into 2 parts. It is for the better understanding. The first part is endergonic pathways. The first part is endergonic pathways. The endergonic pathway, it is clear from the name that the endergonic pathway or endergonic process or endergonic steps require ATP, and it is well known that the 2 ATPs are required in the endergonic pathway of the glycolysis. And the endergonic pathway, we consider the endergonic pathway from glucose to glyceraldehyde phosphate. From glucose to glyceraldehyde phosphate, to the formation of or to the synthesis of glyceraldehyde phosphate is considered under the endergonic part of the glycolysis. And it is well known that at first you remember the steps. The glucose is converted into the glucose 6-phosphate. The glucose 6-phosphate is converted into the fructose 6-phosphate. The fructose 6-phosphate is converted into the 1,6-diphosphate. The fructose 1,6-diphosphate splits into the dihydroxyacetone phosphate and glyceraldehyde phosphate. Again, the dihydroxyacetone phosphate is converted into the glyceraldehyde phosphate. That means the 1 unit of glucose is responsible for the synthesis of 2 units of glyceraldehyde phosphate. Now, if we remember the steps, then it is very easy to remember the name of enzyme. That one, the first is the glucose, and glucose is converted into the glucose 6-phosphate. That means one group of phosphate is added in the glucose. Then where here is the addition of the pyrophosphate, that here one ATP is consumed, and here the enzyme is hexokinase. That means the hexokinase is responsible for the conversion of glucose into glucose 6-phosphate by utilization of one molecules of ATP. The second one is the glucose 6-phosphate is converted into the fructose 6-phosphate. It is very easy. That glucose and fructose are isomer. That means here the enzyme is isomerase, and there is nothing. That means the enzyme isomerase is responsible for the conversion of glucose 6-phosphate into fructose 6-phosphate. Now, the fructose 6-phosphate is converted into fructose 1,6-diphosphate. Here one again phosphate is added. That means here again enzyme is kinase, but it is not hexose, it is fructose. That means the fructokinase. And here one ATP is also consumed. That means the fructose 1,6-phosphate is converted into fructose 1,6-diphosphate by the enzyme fructokinase and utilizes one molecules of ATP here. That means fructose 1,6-diphosphate is present, formed, and fructose 1,6-diphosphate is splitted because it is commonly considered as unstable. And the breaking of the fructose 1,6-diphosphate is referred as aldose condensation. In the term of chemistry or in the term of organic chemistry, it is referred as aldose condensation. And the enzyme, because it is the aldose condensation, the enzyme is referred as aldolase. And the aldolase is responsible for the breaking of 6 molecules glucose into 3 molecules chemical components, that is dihydroxyacetone phosphate and glyceraldehyde phosphate. Then dihydroxyacetone phosphate is converted into the glyceraldehyde phosphate. So, in the endergonic process of the glycolysis, the one molecule of glucose is converted into 2 molecules of glyceraldehyde phosphate by utilization of 2 molecules of ATP. It is the one part of the glycolysis. But one thing important here, which you remember for the NET and other quality examinations, that the one source is also for the animals or for the human beings, that is the glycogen. And here the pathway of glycogen is slightly deviated because glycogen is converted into glucose 1-phosphate. Glycogen is not converted into the glucose, but glycogen is converted into the glucose 1-phosphate and consume 1 ATP. And then glucose 1-phosphate is converted into glucose 6-phosphate. That means here, the first step of the glucose is the conversion of glucose to glucose 6-phosphate is different in the condition of glycogen. In the condition of glycogen, the glycogen is converted into the glucose 1-phosphate, then glucose 1-phosphate is reorganized as the glucose 6-phosphate. Now, it is the part of the first endergonic process of the glycolysis or endergonic part of the glycolysis. In the second part of the glycolysis, we observe that it is associated with the oxidation reduction process. And it is, we all know that in some ATP is synthesized at the substrate level in the glycolysis. And without the oxidation reduction, that is responsible for the change of energetics of the system. The synthesis of ATP is not possible. And here, in the second part, the first important thing is that it is the oxidoreduction process. That means we observe the oxidation and reduction in the second part of glycolysis. Then, at first we again study or we again remember the steps. Then what is the steps? The glyceraldehyde phosphate is converted into 1,3-diphosphoglycerate. The 1,3-diphosphoglycerate is converted into 3-phosphoglycerate. The 3-phosphoglycerate is converted into phosphoenol pyruvate. The phosphoenol pyruvate is converted into pyruvic acid or pyruvate. Then pyruvate is converted into the lactic acid. Pyruvate is converted into the lactic acid. These are the steps. Then first one is the glyceraldehyde phosphate is converted into 1,3-diphosphoglycerate. And here the 1-NAD is converted into the NADH. That is the first oxidoreduction process. NADH2 or NADH plus H plus. That means here hydrogen is released. Then the enzyme is dehydrogenase. The dehydrogenase, glyceraldehyde dehydrogenase. And the activity of the glyceraldehyde dehydrogenase is NAD linked. That means the NAD is responsible for the removal of the protons. Now, the 1,3-diphosphoglycerate is converted into 3-phosphoglycerate. Then, at this stage, one high energy pyrophosphate is removed, and that is received by the ADP, and it converts into the ATP. Here, the pyrophosphate movement of the pyrophosphate group is observed. Then it is here the enzyme is referred as kinase. Now, the 3-phosphoglycerate is converted into 2-phosphoglycerate. And here the enzyme is referred as mutage. Then 2-phosphoglycerate is responsible for the conversion into phosphoenol pyruvate. And here the reorganization of the electrons are observed, which I am just coming on that topic. Here the reorganization of electrons are observed, and phosphoenol pyruvate is commonly considered as the low energetics compound. And the phosphoenol pyruvate is converted into 2-phosphoglycerate is converted into 2-phosphoenol pyruvate in the presence of enzyme enolase. Now, the phosphoenol pyruvate is converted into pyruvic acid in the presence of enzyme kinase, and here again the pyrophosphate group is released and accepted by the ADP. Then the ATP is formed. That means up to the here 4 ATPs are formed. 2 at the 1,3-diphosphoglycerate to 3-phosphoglycerate level, and 2 at phosphoenol pyruvate to pyruvate level. And 2 were consumed in the earlier stage. That means we gain 2 ATP here. Now, the pyruvic acid is converted into the lactic acid. And here, the NADH2 is consumed, which is synthesized during the dehydrogenation of the glyceraldehyde phosphate to the 1,3-diphosphoglycerate. And that is neutralized here. And now the lactic acid is formed. But in the condition when the oxygen is available and the glycolysis is terminated at the pyruvic acid level, then we observe net gain of the 2 NADH2 and 2 ATP. Here, the significant part is also the significant part. First one, the energy is produced here at the 2 level. One, at the glyceraldehyde phosphate to 3-phosphoglycerate level, and second, that is the phosphoenol pyruvate to pyruvic acid level. But the mechanism of the energy production is different. In the first stage, if we consider the non-living condition, then the glyceraldehyde 3-phosphate releases the 12 kilocalorie energy, and 3-phosphoglycerate has 4 kilocalorie energy in the non-living conditions. That the 8 kilocalorie is remain, and that 8 kilocalorie is responsible for the synthesis of 1 molecules of the ATP. And I discussed the same thing. We were discussed the same thing in last class when I delivered a lecture on the coupling reaction. But in the living system, the condition is not same. In the living system, the energy is not released, but energy is transferred in the form of pyrophosphate as a group. That is the pyrophosphate. And that pyrophosphate is released and accepted by the ADP to convert it into the ATP. At the pyruvate level, that is the phosphoenol pyruvate to pyruvic acid level, the electron rearrangement is considered for the conversion of potential energy, obtaining of the potential energy, and that this potential energy is responsible for the synthesis of ATP from ADP. That is the basic, and that is the main point for the energetic level. Now, from this study, we observed that the ATP is used in the cell as the 4. First one, which I am discussing here, that the potential energy used as a group transfer potential. It is not responsible for the transfer of the energy, but the potential energy used as a group transfer potential. It is responsible for the transfer of a group, that is the pyrophosphate. And it is considered that the ATP is used in the cell as in the four form. First, a phosphate donor. Either ATP is responsible for the donation of a phosphate group, or a pyrophosphate donor, or the AMP donor, or the adenosine donor. That is the donor of a group. That is the basic difference of the energetics. Here, again, when oxygen is present, then the pyruvic acid, it is from, we are clear here that the one molecule of glucose, that is the 6 carbon of the glucose, glucose has the 6 carbon, and that 6 carbon is converted into the 2 molecules of the 3 carbon component, that is the pyruvic acid. That pyruvic acid is, if the oxygen is present, then the pyruvic acid is not entered directly in the Krebs cycle, but through the acetyl coenzyme A. It is enters in the Krebs cycle, or it enters in the mitochondria, because all these reactions are occurred in the cytoplasm of the cell. And when the oxygen is present, or when the glucose is oxidatively catabolized, then the pyruvic acid is not entered directly into the mitochondria, but it enters through the acetyl coenzyme A. That means in the presence of oxygen or in the oxidative catabolism, the pyruvic acid is at first converted into the acetyl coenzyme A. And here it is a complex reaction process. That is the pyruvate dehydrogenase system. It is not a single enzyme. But here a system is present that is commonly referred as the pyruvate dehydrogenase system, and it is completed. The synthesis of the coenzyme A is completed in the 5 steps. That means the 5 enzymatic activities or reorganizational activities or reactions are occurred in the synthesis of pyruvic acid to acetyl coenzyme A. And here the enzyme is dehydrogenase, that means it has the NAD linked activities. In addition with the NAD, it is also associated with the coenzyme A, and coenzyme A has the thiol SH. Now the pyruvic acid, acetyl coenzyme A, and NAD linked dehydrogenase, in the 5 enzymatic reactions or reorganization reactions, is responsible for formation of acetyl coenzyme A, 1 molecules of NADH, 2 and 1 carbon dioxide. That means one thing is remarkable here, which you must remember, that 2 carbon of glucose is only entered in the Krebs cycle, not the 3 carbon. Because the glucose is splitted into the 3 carbon 2 pyruvic acid, but only 2 carbon of the glucose is entered into the Krebs cycle. through acetyl coenzyme A. And one carbon of the glucose is released here when the pyruvic acid is converted into the acetyl coenzyme A. Then the coenzyme A is entered. Acetyl coenzyme A is entered in the mitochondrial matrix, where we observed a cyclic catabolism that we collectively refer the Krebs cycle or citric acid cycle or tricarboxylic acid cycle. Anyone. Here, the acetyl coenzyme A acetyl coenzyme A initiates a cyclic catabolism by combining with the earlier synthesized oxaloacetic acid and form citric acid. Why so? It is referred as citric acid cycle, because the Krebs cycle is the first chemical which is responsible for the initiation of the Krebs cycle is the citric acid. But the synthesis is activated by the acetyl coenzyme A. Then again we at first remember the steps that is necessary. The citric acid is converted into cis-aconitic acid. The cis-aconitic acid is converted into isocitric acid. The isocitric acid is converted into alpha-ketogluteric acid. The alpha-ketogluteric acid is converted into succinic acid through succinyl coenzyme A. The succinic acid is converted into fumaric acid. The fumaric acid is converted into the malic acid, and the malic acid is converted into the oxaloacetic acid. These are the cyclic components. Now, if we observe the complete Krebs cycle, the Krebs cycle represents 8 reactions, and the complete process of the Krebs cycle or the complete reactions of the Krebs cycle are associated with the reduction of the NAD or FAD. Basically, they are responsible for the reactions of the reduction of the NAD or FAD. Of course, 1 ATP is synthesized at the substrate level also here. Now, the first step of the Krebs cycle is the synthesis of citric acid from oxaloacetic acid, and that synthesis is associated by the acetyl coenzyme A. And the enzyme is synthetase because it is responsible for the synthesis of citric acid or citric acid synthesis. And here the coenzyme A is released. Now the citric acid is converted into isocytric acid. Here, the name represents that it is the isomeric form. Of course, the citric acid and isocytric acid are isomeric form. But here, the enzyme is not isomerized. Why? Because it is not the isomerization process, but it is the reorganization process. And that reorganization is mediated by the exclusion and inclusion of water, one molecules of water. At first, citric acid forms cis-aconitic acid by the removal of one molecules of water. And then cis-aconitic acid is formed the isocytric or organized to form the isocytric acid by incorporation of one molecules of water. That is the organization and reorganization mediated through the water. So here the enzyme associated is referred as aconitase. Now the isocytric acid is converted into the alpha ketogluteric acid. The process is associated with the NAD-linked dehydrogenase. And here the one molecules of NAD is reduced to NADH2. And one molecules of carbon dioxide is released here. That comes from the glucose. Now, the alpha ketogluteric acid forms the succinic acid. And in this stage, the conversion of alpha ketogluteric acid to succinic acid is a complex process. It is not a simple process, in which the succinyl coenzyme A is also formed. At first, the alpha ketogluteric acid, and here the enzyme is involved, is referred as, because the coenzyme A, I am just asking that, it has the SH group. So here the enzyme is referred as thiokinase. It is clear from the name that one thiol group is involved and one ATP is also released here. That is the thiokinase. And alpha ketogluteric acid to succinyl coenzyme A is associated with the removal of one molecules of carbon dioxide and one unit reduction of the one unit of NADH2. Now, the succinyl coenzyme A is reorganized and gives one unit of ATP at the substrate level. And the coenzyme A group is removed to form the succinic acid. Now, the succinic acid form the fumaric acid. Here, the enzyme is dehydrogenase, but the dehydrogenase is FAD linked. It is not linked with the NADH. And that FADH linked dehydrogenase enzyme is responsible for the reduction of one molecules of FADH. Now, the fumaric acid is converted into the malic acid in the presence of enzyme fumarase, and the malic acid is converted into the oxaloacetic acid in the presence of the NAD-linked dehydrogenase. NAD-linked dehydrogenase. That means, again, the one unit of dehydrogenase is reduced here. to NADH2. That means in a complete cycle, the net products of the Krebs cycle is 2 molecules of carbon dioxide which enters in the cycle through the acetyl coenzyme A, 3 molecules of NADH2, 1 molecules of FADH, and 1 unit of the AT. It is the complete net gain of the Krebs cycle. Just some time before I asked that the fatty acids and proteins or amino acids are also responsible for the oxidative catabolism. That means after the enzymatic activities, the fatty acids enters in the Krebs cycle through the acetyl coenzyme A by the method of beta oxidation, which we will discuss in further class. Second one, the breakdown of the amino acids, and the amino acids 20 types. We know very well that the 20 types of amino acids, and the entry point of the all amino acids are not same in the Krebs cycle, and that may be asked in your examinations. Commonly, out of the 20, 10 amino acids enter in the Krebs cycle through the acetyl coenzyme A, but the remaining amino acids are not entered through the acetyl coenzyme A, but at any stage of the Krebs cycle, which is necessary to remember. And I am telling the name, I am here telling the name, and some biochemical methods are associated with the breakdown of the proteins, which again we will discuss in the coming classes. The 5 amino acids, arginine, histidine, glutamine, glutamic acid, and proline. I am again asking, arginine, histidine, glutamine, glutamic acid, and proline are entered in Krebs cycle through alpha ketogluteric acid. 3 amino acids, that is isoleucine, methionine, and valine. That is isoleucine, methionine, and valine entered in Krebs cycle through succinyl coenzyme A and asparagine and aspartic acid enter in Krebs cycle through oxaloacetic acid. That means the energy produced by the oxidative catabolism of amino acid are not same, and they vary from amino acid to amino acid. That is the question. And you must analyze the production of energy with respect to the particular amino acid through the Krebs cycle. Now, the reduced carriers or coenzymes or factors, that is the NADH2 or FADH, produced or released by the catabolic reactions of the carbohydrate, of glycolysis or Krebs cycle are carried to the to a system which is responsible for the transfer of electron from higher energetic level to lower energetic level by the process of oxidoreduction method. And this process is referred as electron transport system. Then the electron transport system is arranged sequentially from the higher to lower energetics level as flavoprotein. Flavoprotein is followed by coenzyme Q. Coenzyme Q is followed by cytochrome B. Cytochrome B is followed by cytochrome C1. Cytochrome C1 is followed by cytochrome C, and cytochrome C is formed by cytochrome A1 plus A3. Here, the sequences are present in the form of higher energetic value to the lower energetic value. The first, we observe that there are 5 types of cytochromes are present, containing heme group, that is the iron group, which undergoes reduction and oxidation reaction, in which the A1 plus A3 both collectively are collectively called cytooxidase or the respiratory enzyme. That is the important question. The respiratory enzyme term is used only for the A1 plus A3 cytochromes, not for all. And coenzyme Q is chemically ubiquinol. It is chemically ubiquinol. Second one, flavoprotein. Flavoprotein are enzyme containing flavin coenzyme and riboflavin 5-phosphate. That means we use the riboflavin, a type of vitamins, pyridoxine, a type of vitamins. You must remember all these vitamins. Now, at the A3 unit, one important thing is here. At the A3 unit, the copper atom is present, and the copper atom of the cytochrome A3 unit gives characteristics electron spin resonance signals. It is only provided by the copper atoms and undergoes cupric to cuprous transition during electron transport and finally released to electron or H plus to oxygen and form the water. That means the pair of electron derived from the catabolic process during the glycolysis or Krebs cycle flow down a multi-membrane chain of electron carrier enzymes of successive lower energetic levels and release the energy, that is the free the energy in the system. That means electron transport system is not responsible for the synthesis of the ATP. It is only responsible for the release of energy to the system when electron moves from the higher energetic value to the lower energetic value. And the energy present in the system, and particularly in the mitochondria, inner wall of mitochondria, that is at the cristae, the enzyme is used for the conversion of ADP into ATP, that is synthesis of the high energy pyrophosphate and its association with the ADP. And this process is referred as oxidative phosphorylation. That means the electron transport system and oxidative phosphorylation are different things. Or in other way, it is better to say that the release of energy and synthesis of the ATP are different. Of course, the energy released in the system by movement of the electrons from higher energetic level to the lower energetic level is responsible for the conversion of the lower energy phosphate bond to the higher energy phosphate bond and synthesis of the ATP. But both are different, both are not same thing. In this way, we observe the catabolic actions of the glucose, which is primarily responsible for the source of energy of all living systems, except the autotrophic plants. And here, again, the complete process of the oxidative catabolism of glucose is occurred in mitochondria, in eukaryotic cells, and at the cell membrane as the prokaryotes. Earlier mesosomes were assigned for the what? But no, the complete membrane, cell membrane of the prokaryotic cell is responsible for the production of energy by the oxidative catabolism of glucose. Now, I am giving a brief idea of the catabolic activities, and I think that you have no more problem in the related topics. And further, one part of the energetics and catabolic reactions associated with the fatty acids and amino acids are remained, which we will discuss in the coming class. And it is necessary to say that the students associated with the or attached with in the live classes must prepare for the NET and other related examinations with full honesty. Thank you. We will meet in the classroom very soon. Very soon we will meet in the classroom. Thanks to all for the live association. Thank you.